is the first public park in Putrajaya. It was opened in 1st February 2001 and launched on 2nd February 2001. This is the largest man-made wetland in Malaysia and tropical Asia, with 24 wetland cells that clean water from rivers before entering Putrajaya Lake. The wetland plays a big role in keeping the environment clean and balanced. It also has a nature interpretation center for fun and interactive learning. During our visit, we were given a task to find and identify several types of grass that are useful for the lake. These grasses help filter water, support biodiversity, and keep the lake healthy. So Wetland Park is not only beautiful, but also full of knowledge and exciting activities. Moving on to Putrajaya Botanical Garden. This is also known as the mother of all gardens. It was opened in 2003 by Tun Dr. Mahathir Mohamad on land that was once used for rubber and palm oil plantation. The garden symbol is a rare palm tree called Johannes Dagismania parenthesis, which takes 30 years to flower. This garden is a center for research, conservation, and education. During our visit, we did two practical activities. The first was about the basic concept of statistic and variation, and the second focused on the factors that affect the distribution and abundance of organisms.
Practical 1. Basic concept of statistics and variation. In this practical, we have learned about normal distribution and its parameters, the mean and variance, and initial understanding about correlations. The objectives of this practical are as follows. The first one is to calculate and describe the following descriptive statistics, mean, variance, and also the standard deviation. Secondly, to understand the concept of correlation. And the last one is to determine the relationship between the two variables. In practical 1, we have collected 3 sets of data, set 1, set 2, and also set 3. So for each of the sets, we have calculated the mean, variance, and also the standard deviation by using the formulas that have been stated in our lab manual. So moving on to the first set, which is set 1. So we have two different kind of tables, which is the leaf length in cm and also the leaf width in cm. So in order to get the mean, we have to sum up all the leaf length in cm divided by the total leaves, which is the n. So for the mean, we got is 13.5. Moving on to the next column, which is the variance. In order to get the variance, we have to square the mean and minus 1. So our variance is 0.84. Next, the last column is the standard deviation. For the standard deviation, we have to square root the variance, and the standard deviation that we got is 0.917. Next, for the leaf width in cm table. So in this table, we just use the same formula in order to get the mean, standard deviation, and also the variance. So for the mean, we got is 5.47. For the variance is 0.2179, and for the standard deviation is 0.4668. Then moving on to the second set, which is set 2. So for the first table, for the leaf length in cm, for the mean, we got is 13.48. For the variance, we got is 3.008, and for the standard deviation that we got is 1.734. Next for the leaf width, the mean is 5.02, the variance is 0.4062, and the standard deviation is 0.6374. Last but not least, set 3. So for the first table, the mean is 12.22, the variance is 1.7507, and the standard deviation is 1.3231. And for the last table, which is the leaf width in cm, the mean is 5, the variance is 0.1289, and the standard deviation is 0.3590. So the mean length for the set 1 and also the set 2 are very close, 13.50 cm and also 13.48 cm, indicating a high similarity in leaf length among those two sets. However, set 3 shows a slightly lower mean, which is 12.22 cm, shows that the leaves in this group are generally shorter than those in the other two sets. So the leaf width shows more variation among the sets compared to the length. Set 1 has the highest mean width, which is 5.47 cm, while set 2, which is 5 cm, has slightly higher mean width than set 3, which is 5.02 cm. So this indicates that leaves in set 1 are generally broader, while leaves in set 2 and set 3 are narrower. So this difference in mean for each set could be due to environmental factors such as sunlight, water availability, and temperature, or variation in leaf maturity of health. So in conclusion, set 1 and set 2 exhibit similar average leaf length, suggesting consistent growth, while set 3 has shorter leaves. So for width, the set 1 stands out as broader, while set 2 and set 3 are quite similar in leaf.
Variation. So first of all, variance is a statistical measure of how much the values of a dataset differ from the mean of the dataset. So variance shows how spread out the data is. So the statement shows that each leaf has different measurements in length and width. So in terms of variance, it means that there is a variability in the data collected. So to obtain variance, so value of mean must be calculated first. So if the leaves have a wider range of sizes among the leaves, so high variance value is obtained. If most leaves are close in size to each other, lower variance value is obtained. So standard deviation is a statistic that measures the dispersion of a dataset relative to its mean and is calculated as the square root of the variance. So in conclusion, based on the statement, each leaf size measurement is not uniform, and the variation in size is proved numerical by variance.
It's necessary to have different sets of measurements for the measurement of leaf width and length to obtain more accurate results. It helps determine whether different plants share similar characteristics. This detailed data enhances our understanding of plant traits and variability. Distinguishing between width and length provides a detailed view of leaf morphology. It contributes to knowledge about plant diversity, adaptation, and ecosystem roles.
It's practical 2, factors that affect the distribution and abundance of organisms.
In this practical, you will explore how to study plant communities using the quadrant method. You will learn to observe and record both biotic and abiotic factors, identify plant species, and compare population sizes to understand how organisms interact within a habitat. The objectives of this practical are as follows. First, to introduce the simple concept of quadrant. Second, to enhance the power of observation. Third, to record the difference in plant distribution within the quadrant. And lastly, to infer from natural observations the possible factors that affect distribution and abundance of organisms.
Each of the following species of Calophyllum in Ophyllum, Hopia odorata, Dipterocarpus oblongifolius, Deloniceregia, and species 1 was represented by only one individual. To calculate the density, we use the formula density is equal to the number of individuals of a species divided by the total area of the quadrant. Since the quadrant measured 5 meters by 5 meters, the total area was 25 square meters. Therefore, the density for each of these species is 1 divided by 25 square meters, resulting in 0.04 individuals per square meter. In contrast, there were 2 individuals of Arenga hookeriana found within the quadrant. Using the same formula, the density is 2 divided by 25 square meters, which gives a result of 0.08 individuals per square meter. Each of the following species of Calophyllum in Ophyllum, Hopia odorata, Dipterocarpus oblongifolius, Deloniceregia, and species 1 was represented by only one individual. To calculate the density, we use the formula density is equal to the number of individuals of a species divided by the total area of the quadrant. Since the quadrant measured 5 meters by 5 meters, the total area was 25 square meters. Therefore, the density for each of these species is 1 divided by 25 square meters, resulting in 0.04 individuals per square meter. In contrast, there were 2 individuals of Arenga hookeriana found within the quadrant. Using the same formula, the density is 2 divided by 25 square meters, which gives a result of 0.08 individuals per square meter. Next, for the observation at Site 2, which is the quadrant located in the middle of the forest, we identified 7 different plant species. These species are Alstonia angustinoba, Dipterocarpus oblongifolius, Paricia insignis, Cartophyllum fragrans, Salaka zalaca, Liquala spinosa, and one unidentified species referred to species 2. In this observation, each plant species was represented by only one individual. The formula used to calculate the density is the same as in Site 1, where density is defined as the number of individuals of a species divided by the total area of the quadrant. Since the quadrant measured 5 meters by 5 meters, the total area was 25 square meters. Therefore, the density for each of these species is 1 divided by 25 square meters, resulting in 0.04 individuals per square meter.
It is an ecological sampling unit consisting of a small square area of ground within which all species of interest are noted or measurements taken by having methods that count all individuals on several quadrants of known size, then extrapolate the average count to the whole area by using diversity index or relative density formula. Area of the quadrant must be known and quadrants must be representative of the area, which are achieved by random sampling.
The advantage of larger quadrat is they can include more individuals and species, which offering a better representative of biodiversity. Next, however, the disadvantage of larger quadrat is they are more time-consuming, harder to manage, and may increase the likelihood of human error. Additionally, species distribution in large habitat is often unevenly spread, leading to unrepresentative sample in execution.
The difference in the number of plant species within the quadrant likely occur due to microhabitat variation and differences in environmental conditions that affect plant distribution. Additionally, human disturbances such as trampling, pollution, or age effects may contribute to patchy distributions. These combined factors result in a non-uniform plant species composition across different quadrants.